My name is Josh LaGrange, and I'm in the HYRS 2010 group here at the U of A this year, and I work with Dr. Andrew Simmons. My lab studies RNA processing bodies, or P-bodies. RNA is a messenger molecule that carries genetic information from the DNA and uses it to make proteins. It was once thought that gene expression was controlled simply by turning genes on and off, but then it was found that some RNA is degraded after it's produced, silencing the gene it encoded. And about 8 years ago, it was discovered that this RNA degradation takes place in specific sites in the cell, which are the P-bodies. My lab attempts to gain a solid understanding of these P-bodies. I study a protein called Pac-Man, one of the proteins inside the P-body that degrades RNA. To study it, I attach a fluorescent protein from jellyfish to Pac-Man. I then put this into fly cells and observe the protein's location and movement throughout the cell using a microscope. I observe Pac-Man in conjunction with other proteins with different colored fluorescent tags. If two proteins end up in the same place, or colocalize, I can conclude that they work together. The rest of my lab experiments with various proteins found in P-bodies using the same methods to gain a better understanding of all the proteins involved. With the knowledge gained from these experiments, we hope to gain insight and understanding into the very lethal diseases that are associated with abnormal RNA degradation. Hopefully, with this better understanding, we can develop treatments for these diseases.